So we'll start with the next chapter, that is, we have already talked about the sericulture, morphology, then we talked about the different mulberry trees and different strains of the silkworm that we get. That is, we talked about the mulberry, then we talked about the muga, eri, and tussar silkworms. Today we'll be talking about the silkworm biology, that is the morphology part of it, what they are, then what are the different systems that they have in them, how are they going to help them, and of course not to forget the silk gland, which is most important because that is why we are culturing this silkworm to produce the silk. So when we are so much interested in the silkworm, the silk, we should know about its biology too, so that we can take good care of them, and by so they also give us a proper silk, which is of a higher quality, and of course they are useful in various ways. Rather, they are used for parachute making, they're used for military services, they're also used in medical services where these threads are used as sutures to stitch it, because earlier we had those thread system where the doctor had to cut the threads after the operation, but now we are having this silk threads used as sutures to so that after the suturing, the doctor doesn't have to cut them, because the silk dissolves on its own internally. So these are some advantages of using other than the fabric. So it is our duty to understand the biology of the silkworm so that we can have a better understanding of the silk worms, the silk insects, the larva. Okay, so let's start with the morphology of or the life cycle of the silkworm. So for example, first you can see the picture over here, how the life cycle occurs. We have already talked about this life cycle when I was talking about Bombyx mori in the first class. Nevertheless, this is the adult male and the female laying eggs, and then these eggs are hatched to form the larvae. So you have 5 instar larvae on this, and then the larvae, after having its intake, a proper intake, that is, as we talked about them, they are voracious feeders. After eating, the 5th instar larva forms a cocoon, that is the pupate inside, called as chrysalis, and during this period, the silk thread is woven around them, and this is a cocoon that we are interested in. And if the silkworm is not removed, after some days, the adult emerges from the cocoon. So what is the life cycle, and what are the different stages, how they look like, is what we are going to talk about in the silkworm. So let's start with the first one, that's the life cycle. So you can see over here, after the adult have hatched an egg, the egg either undergoes hibernation. That is, hibernation is a diapause stage where the egg take a sort of a interval or a resting period. Because why they have to undergo a resting period is when the climatic condition is unfavorable. For example, if it is too cold during the winter season, the silkworm do undergo hibernation, and sometimes they do not undergo hibernation, so directly they go under hatching. At this point here, what we can do is we can also induce them artificially to hatch them if we want to speed up the process, because hibernation can take place for months together, which will delay our production of the silk. So artificial induction is done so that the eggs hatch immediately. And once the egg hatches, it goes into the, it's called as a first instar larva. The first instar larva, through molting, undergoes 4 molts. And then the last instar larva is what you call as the 5th instar larva, which is a mature larva. And during these instar larvae, they, as we said, they are voracious feeders. That is, they just eat, and eat. So that is the only thing they do. because as they are eating, the production of the protein increases in them through the mulberry leaves. The important proteins called the fibroin and the sericin is produced as they are feeding on the mulberry leaves. And this is what is spun out in the form of silk threads around the pupa. So that's called as a silk thread or the silk of the pupa. So after they have had enough food, the fifth instar larva slows down, they stop eating, and they undergo pupa formation or metamorphosis. So as they are pupating, they secrete the silk gland, which is woven around them, and then they form a pupa. And from the pupa, that is after the metamorphosis occurs inside the pupa, an adult emerges out after some days, and that adult, they survive only for 3 or 4 days. And within those days, they lay their eggs, and very sadly, they die. So that is the life cycle of the mulberry silkworm. And sometimes they complete within 45 to 60 days, or sometimes a little bit earlier if it is a multi-voltine. It can occur within 20 to 25 days. If it's a bivoltine or a univoltine, that is when the life cycle is completed in 1 year or say around 8 months or so, then the silkworm life cycle increases. But in a multi-voltine, it can complete within 20 to 25 days. So it consists of the stages of egg, larva, pupa, and a moth is what we're talking about. And the egg stage lasts for around 9 to 10 days, whereas the larval form is for up to 25 to 30 days. So this 25 to 30 days is what we are talking about in a multi-voltine. So what do you mean by a multi-voltine? The life cycle is not only once, it can be 3 or 4 times. They complete in 1 year, 3 to 4 times of life cycle occurs. So that is called a multi-voltine silkworm. And the pupa stage goes around 10 to 15 days, and a moth stage is just 3 to 4 days is what we are going to talk about. So when we are talking about the morphology of the Bombyx mori, so we'll have to talk a little bit about the eggs. So what are the eggs? First thing is the silkworm passed through a complete metamorphosis. So what do you mean by a complete metamorphosis? It has an intermediate stages of larval form and a pupa form. So a complete metamorphosis occurs in them. And after the complete metamorphosis occurred, adult moth emerges out from the pupa. And the moment the adult emerges from the pupa, within 3 to 4 days, they are able to fertilize their eggs, copulate, fertilize the egg, and they're going to lay their eggs. The egg are very tiny. If you can see over here, they are small, tiny white color eggs that are laid. So when you are culturing, we try to take the female and cover it in a small cup around them, so that the moth lays its egg in a circular motion. So you have a nice round area where you can take care of the eggs over there. So these eggs are very tiny. That is after every 200 grams, 2000 eggs per gram laid, you can count their weight. That is, each single egg is going to be around 0.5 to 0.6 mg. So that tiny they are in them. And if you can see the egg over here, they have a nucleus. The dark dot is a nucleus, and you have the periplasm over there inside. That's an anterior pole on top and a posterior pole below, with a micropyle on top over there. They have a outer covering that is a chorion. Then they have a inside vitelline membrane and the yolk substance that is present over here for the nourishment of the growing zygote. So that's the structure of the silkworm. The larval forms over here, if you can see, the silkworm larva when newly hatched is a black or dark brown color. So you can see those nice tiny blue colored eggs over there from where the small silk larva emerges. And once they emerge, they resemble like that of a small ants over there. and they have 4 pairs of tubercles. There's the subdorsal, supraspiracle, infraspiracular, and basal tubercle, each of which carries 3 to 6 setae. And the entire body, so if you can see over here, the entire body over here is covered with a thin and elastic chitinous cuticle, which is capable of being extended considerably to permit rapid growth of the larva during any instar. So the entire body that is covered over there is of some use over there for them to that is because they have this thick and elastic chitinous cuticle, which is capable of being extended. So what happens is during every molting stage, they're able to grow in size, because molting in any larval forms means because the larva doesn't grow in size as such. So they have to mold. That is, they have to remove their old skin and they have to go into the next instar larva is when they start growing. So that is the most important part over there. And so this is how a larva, the 5th instar larva looks like. So here you can see they are very tiny ant-like structures coming out of that small egg, and you can see those the spiracles, the cuticles present over there, which helps them in growing them. And if you can see in this picture, this is the 5th instar larva, that is the larger larva of considerable size over there. And also the thickness also increases. The larva of the silkworm is divided into head region, the thoracic region from this part to this part over there, and from this part over here till the tail region is the abdominal region. So what are they? The head region consists of the mouth parts, whereas the thoracic region is divided into 3 segments. That is the prothoracic region, which is containing the first spiracle over there. Then it has a mesothoracic region and a metathoracic region. So this the thoracic region is divided into 3 regions over there: prothoracic, mesothoracic, and metathoracic region. And in the first thoracic region, you have the first spiracle. Then comes to abdominal region. The abdominal region is made up of 9 segments over there. So the second spiracle starts from the first segment of the abdomen over there. So you have those 9 segments with the 9 spiracles present over there. Then you have a caudal horn over there. So that's called a tubercle horn. The body has, as we said, they have 3 pairs of tubercles. So here we are saying, sorry, they have 4 pairs of tubercles, which helps them in movement as such. So here you have those movements. These tubercles are sometimes, they behave like that of the bony structure that you find on the larval form. And this bony structures are sometimes this can mimic the bony structure of the vertebrates, where you have some bony structures at the cartilaginous area and also in the mucous area. So somewhat protection that you find in this particular larval forms. So you have those 2nd, 3rd, 4th, 5th, 6th, 7th, 8th, 9th spiracle over there. And at the end, you have a caudal horn and the anal papillae. And of course, you can find a caudal leg over there, 1 pair of leg over there. And you have 4 pairs of leg at the abdominal segments, that is from the segment 6th to 8th over there. And then you have those thoracic legs over here, 1 on each of the thoracic segment over there. So these thoracic legs are the true legs that will be formed when the larva undergoes metamorphosis. These legs are the one that is going to stay back, whereas the legs that you find on the abdominal and the caudal region is not going to form any legs. They are just false legs. So this is the structure of the larva that you will have to talk about. Okay, coming to the head part of the larva. So here you can see this is the small head part of the larva is what we are going to talk about right now. So this is the head of the larva. And why we are going to have a direct focus on the larva is we want to talk about the mouth parts of the larva. So what are the different mouth parts of here? The head consists of 6 body segments. These are the 6 body segments that you are going to talk about fused together in the cranium. The cranium is the head region. The 2nd, 4th, 5th, and the 6th segment carry appendages, which are modified into the first part. You call them is the antennae. Antennae are sensory in nature, tactile in nature. Then you have the mandible over there on either side. So whatever I'm talking about, they are in pairs over here. So you have a pair of antennae on either side. This is one antennae over here. This is one antennae over there. And you can correlate these two antennae that is present over here. So these are not the eyes that you're thinking about, but they are the antennae, which are sensory in nature. Here you have those ocelli, three pairs of ocelli on each side. One this side and one that side, the dark dots over there, ocelli, which again acts as a sensory in nature. And then you have a mandible, a pair of mandibles. So this is a mandible on either side. And if you can see over here, those denture-like structures that you find over there. So these are the mandibles that you can see over there, which help them in mastication of the leaves. So whatever the food they are eating, they have to masticate and take it in. So this mandibles helps in mastication of the mulberry leaves. Then below that, you have this maxillary lobe. This is the maxillary palp. Underneath is a maxillary lobe. So this is a maxillary that you find on either side. And in between, you have a labium. And the labium consists of a labial palp on either side. That small extensions over there is the labial palp. And this, the center one, is a spinnert through which the larva spins the cocoon formation. That is, the silk is silk gland secretes the silk around to form the cocoon over there. So that is a spinnert over there. So here, when you're going to talk, this is the labrum, that is the topmost part of the mouth parts over there. So this is the labrum over there. These two are the mandibles over here. These two are the antennae over there. Then you have the labium that is present over here with the maxillaries on either side. Then you have the small, tiny labial palp on either side. And in the central region, you have that spinnert, which helps in secretion of the silk gland. So they help in the secretion of the silk gland from the mouth region. So this is the head region of the larva. So here you can see these two dots are not the eyes, but they are the antennae of the head region of the larval form. Okay, coming to the thoracic region. So thoracic region, as I've already told you, it consists of three segments: pro, meso, and metathoracic region. And ventrally, there is a pair of leg, each pair of leg on each segment, which helps in the movement of the larva. And of course, they are the true legs because they are conical, and they are comprised of three jointed segments. So each pair of leg is comprised of three jointed segments. And this is what makes them come under the phylum Arthropoda, because they are all insects. They come under phylum Arthropoda. The abdominal region of here, so what you can see in the abdominal region, they are composed of 11 body segments. But when you count, you get only 9 body segments, because the 9th one is fused with 3 segments over here. And hence they look as though it is only a one part, but they are fused at the 9th region with a caudal leg on the ventral part and a caudal horn on the dorsal part of the body over there. So they are made up of 11 segments. That is, 9 can be distinguished as 3, that is the 3 pairs that is fused over there. So for example, you have 3 segments at the thoracic region over there. Then the rest is the abdominal segment, that is the 9 segments, but they are not 9. The 9th one is fused with 3 segments within inside. So that is the abdomen. And on the abdomen, you can find there are some respiratory pores, or the spiracles that you find over here. So these are the spiracles which helps them in respiratory process. So that is one spiracle is on the thoracic, the first thoracic region. The rest all you find them on the abdominal segments. So that is the abdominal region over there. Then when you're talking about the pupa stage, that is after the larval formation, the 5th instar larva, after each molting over there. The pupa stage is generally called as a resting stage, because they need to rest. The larva, after eating voraciously, they need to rest for some time. Why? Because they have to undergo metamorphosis. So the complete change of the body structures, it has to grow its wings, it has to grow its appendages, it has to grow its antennae, and the abdominal segments have to be well developed over there. So they have to have a resting stage or inactive stage, what you call them. This is a stage where they do not eat anything over there, the fifth instar larva relaxes itself, and is when the pupa formation is occurring. So they are called as inactive stage of the silkworm. And soon after pupation, the pupa is white in color and soft, but gradually turns dark brown. Can you see the difference over here? So this is the first stage of the larva when they have undergone pupa formation. So they are white in color, but as gradually the metamorphosis is occurring within inside, the pupa slowly turns its color from brown to dark brown in color. And prominent morphological parts are visible as a pair of large compound eyes, a pair of antennae, fore and the hind wings, and the legs can be clearly seen within the cocoon formations over there. Okay, so that was about the pupa. So here you can see, you can differentiate the male and a female pupa as they are in the pupal stage itself. So you don't have to separate them when they become a moth. So how you are going to identify whether it's a female moth or it's a male moth? If you can see over here, in a male genital marking, so you have on the 8th abdominal segment, you have those genital marking in the pupal formation, whereas in a female, you have a X-shaped mark over there, which is not found in the male over here. So a fine longitudinal line on the 8th abdominal segment, whereas such a marking is absent in the male abdominal region. And of course, the abdomen is broader in the female, and the abdomen is a little bit narrower in the male pupa formation. Although it is very difficult to differentiate the broadness of the abdomen over there, the best thing is to check that fine longitudinal line that you find on the 8th abdominal segment to demarcate between a male and a female. Okay, so how is the adult going to look like? So you can see after the metamorphosis has occurred, the adult emerges from the pupa over there. So they have those wings, those antennae, the abdominal segments that they are found. And you can also see that in the male is always narrower or thinner when compared to the female over there. And what is the other important feature of there? You can see the antenna over there. The antenna is longer in the males, whereas the antenna is smaller in the female. So this is how you are going to differentiate between a male and a female moth. And these are unable to fly. They don't fly at all. As you can see, the wings also, they are very small wings. If you compare that site over there, this is only a diagram over there, but if you can see the live or the real pictures of the moth over here, the wings are very small. Why? Because over the years, because they were domesticated for culture of silk, the silkworm culturing, because they were domesticated, they never had to fly in the wild. So they almost forgot flying, rather you can say. So they don't fly much. And of course, because they don't feed during that stage, because they completely stop feeding, their work is only to copulate and lay eggs. So it's a very short duration of adult life, probably just 3 to 4 days of time. So the body of the moth is composed of 3 distinct segments. You can see of the head region, the thoracic region, a short thoracic region, and an abdominal segment. And of course, what I said over there, the female has a smaller antennae, whereas the male has a larger antennae, is how you're going to differentiate between a male and a female moth. Okay, now we'll talk a little bit about its systems. We'll be talking about the digestive, respiratory, circulatory, reproductive, excretory system over there, because you have to understand what exactly their uses are over there. So if you can see in this picture over here, the digestive system is a long elongated structure over here, divided into foregut, midgut, and the hindgut over there. So it's more or less a straight tube from the mouth region over there to the anus region over there, and divisible into stomodium or the foregut, the middle mesenteron, whereas the posterior region is also called as a proctodium. And the foregut consists of a narrow pharynx, which opens into a small esophagus, and the esophagus opens into the midgut, larger midgut over there. And between the foregut and the midgut, there is a cardiac stomodial valve, cardiac the heart region over there, a stomoidal valve. And the midgut is long, wide cylinder tube and narrow at the posterior region. So they are wider in the midgut region and they slowly narrow down at the posterior region. And they are protected by peritrophic membrane from the inner side. Okay, so how do they look like if you're going to take up the lateral section of the larval form over there? So you can see over here. So this is the mouth region over here, and we have already discussed what is a labium, what is a labrum. The labrum is the upper covering of the mouth. The labium is the lower part where the salivary gland is situated for the secretion of the saliva over there, the labrum. And it opens into a mouth. The mouth opens into a pharynx, a short, thin pharynx over there. There's a hypopharynx below that. The pharynx opens into a esophagus, and the esophagus opens into a larger gut, or the crop region that you call over there. And the gut region opens into the, so this is the foregut, and the foregut opens into a larger, wider midgut that you call the middle part of the stomach over there. And the midgut opens into a posterior hind gut over there. So this is the hind gut over there, which opens, and this hind gut opens into the anterior intestine. The intestine opens into the rectum over there. So this is the rectum, and then it opens into the anus. So this is the digestive system. A straight tube is what we're going to talk about in the silkworm over there. So the pylorus valve is situated between the midgut and the hindgut. So there is a pylorus, the midgut, and the hindgut. So here you have a pylorus or a pyloric valve. The hindgut consists of a small intestine, colon, and rectum. And the rectal is provided with the rectal muscles, the rectum over here. So here is the rectum over here. The rectum is provided with the rectal muscles for pressing. That is, the muscle contraction and expansion helps in removing the excreta for excretory purpose. So that is the most important function of the rectal muscle in the rectum region. So what happens? Digestion and assimilation of the food occurs in the midgut. So here in the midgut, the digestion and assimilation. What is assimilation? To assimilate or to absorb the digested food, because whatever is eaten has to be digested, and whatever has been digested has to be assimilated by the body or absorbed by the blood cells, so that it can be given to different parts of the body. The nutrition and the oxygen carried to different parts of the body. So assimilation of the food occurs in the midgut, while the hindgut absorbs excess moisture through rectal muscles. So whatever the moisture that is present, because these insects, they don't drink water, or water is not available for them. So whatever moisture is present in the leaf that it has been taken, they have to protect them. And so they don't tend to throw them out, is what they are mostly absorbing back the moisture inside the hemolymph, and the undigested food is eliminated from the anus region. Okay. So what is an excretory system? So we talked about the digestive system. We'll talk a little bit about the excretory system. Excretory system in insects, you will always talk about Malpighian tubules. Mostly in the insects, those which are having the wings, they have this Malpighian tubules for excretory purpose. And they arise at the midgut of the small intestine. So they arise at the midgut of the small intestine. If you remember that the digestive system over here, so this is the small intestine, is where they arise over there. So you see that Malpighian tubule that is present over here. So this side and that side, they have those two Malpighian tubules at the intestinal region over there. So they arise at the junction of the midgut, small intestine, and colon in the hindgut. So this is the colon in the hindgut region and the midgut region over here. So they arise at this point somewhere over here. The tube arising on either side is enlarged into an excretory chamber called the urinary bladder. So this is the urinary bladder from which two branches arises. So the tube arising on each side is enlarged. The tube arising on each side is enlarged over here. And this enlarged is the excretory chamber or urinary bladder from which two branches arises. So this is the two branch that you can find over here. And one tube situated on the dorsal side branches further into three branches. So you can see one branch over there, you have one branch over there, and you have one branch over there. So you have 3 tubes that are arising from this particular tube over here. So which has branched over here. So they are going to divide into 3 branches, one this side, one this branch over there, and one on the other side. So you have 3 branches over there. And all the 6 tubes. So what is a 6 tube? We were talking about 3 tubes. Then from where did the 6 tubules come? So if you count over there, each tubule having 2 branches, 1, 2, 3, 4, 5, 6. So these are the 6 tubules. So this 6 tubules stick to the midgut. So you can see this is the midgut over there. The larger, wider part of it is the midgut. These tubules stick to the midgut and run towards the anterior region. So they are running to the anterior region, and immediately they take a U-turn and run posteriorly downwards. So they go on top and then they come down, they go on top and then they come down. So as they are coming down towards the posterior side, they ultimately open into the rectum. So this is the rectum region over here, where they ultimately open into the rectal region. So you can see one tubule, one part of the tubule over here, one over here, one over here. So all these ultimately open into the rectum. And this condition is called as cryptonephridial condition. So what is a cryptonephridial condition over here? This cryptonephridial condition is adapted for arid region, that is in the dry region, where they have to absorb maximum moisture content present in the undigested food, so that the moisture is retained in the body. So that's a cryptonephridial condition over there. So what is happening over here? The tube passing towards the anterior region, they have to, those that are passing, so what the picture that we are seeing over here is the same as that over here. So as the tubes are moving on top, the movement on top is the ascending tubes, and that comes back down is a descending tube, is what is talking about over here. The descending limbs, ascending limbs or the descending limbs. Those that moves up is an ascending one. Those that comes down is a descending limbs. And what they do, the nitrogenous compound like proteins are metabolized by these tubes and excreted principally as uric acid. So the proteins, because as we said, the leaves contains lots of proteins in them. So the proteins have to be metabolized. And after metabolism, the end result is a uric acid, which has to be excreted out. They also excrete calcium oxalate, a little bit of them. And the Malpighian tubules, tubes in the mature larva are light yellow, and their walls contain large quantities of yellow vitamin B2. So what is this yellow vitamin B2? Do we have vitamin D2 in our body? Yes, we do have vitamin D2, B2 in our body. Why? Because they are helpful for growth factors, for example, like for metabolism of the proteins and fats, this particular vitamin B2 is needed. For the growth factors, they are needed, and they're also needed for, they help in reducing the oxidative stress in the system. Oxidative stress is if there is more amount of peroxides or oxides that are present in the body, they induce oxidation stress in the body. They have to be removed time and again. So this vitamin B2 helps in that. They also reduce the inflammation that is occurring in the nerves. So they relax them, and then a smoother movement or smoother development of the body occurs. So, and of course, as I told you, they also are needed in the metabolism of the proteins and fat. If vitamin B2 is not present, then the proteins will not be metabolized thoroughly, and the uric acid will not be excreted. If the uric acid stays back in the human body, what happens is they form gout, that is small bone enlargement occurs in human beings. So we are talking about the human beings, but here, when you're talking about the vitamin B2 importance, these are necessary for metabolism of proteins and fats in the larval forms. Okay, so that was about the excretory system. We'll talk a little bit about the respiratory system of the silkworm larva. So what is the respiratory system of there? As we talked about the larval form, we talked a little bit about the spiracles that are present in the first thoracic region and the rest on the abdominal region. So here you can see So these are that small spiracles, the round small spiracles that you find over here, the dots over there. So these are the spiracles that you find on the abdominal region, and one at the first thoracic region over there. And these are connected by a tracheal trunk, the main tracheal trunk. So this long extended one from the mouth to the anus, completely at the end of the caudal segment over there. So you have that main tracheal trunk, and this tracheal trunk gives out tracheal bushes that you can see over there. So these are some bushes that you can see, which are extending onto the body surface of the larval form over there. And this tracheal bush is the one that becomes tracheoles for delivering the oxygen to different parts of the, taking the oxygen from different part and giving it to the blood supply over there. So every part over there, tracheoles, they take in the oxygen over there. So when you're talking about the spiracle, one small spiracle, so this one small spiracle over here, if you can check it up in an enlarged form, they are covered by a peritreme over there. So this is an outer peritreme, and this is an opening through which the air is passed inside. And this is a sieve plate. Sieve plate is what is called something to filter. So sieving is to filter. And what is the function of a sieve plate? To filter the dust particles, which does not allow them to enter inside, because they are having so many spiracles. The chances of dust particles, like what we have in our nose, we have those small hair which protects us from the dust particle to enter inside. So the sieve plate over here is doing that same work what a nostril does for us. Okay. This respiratory function is carried out through spiracles by the movement of body walls. And as I said, there are 9 pair of spiracles, one on the first thoracic region and the rest on the abdominal segments. The peritreme, sieve plate, and the atrium are the parts of the spiracle. So these are the parts of the spiracles. And the peritreme forms a ring around the spiracle, and a slit is seen in the median line of the sieve plate inside the ring. So what is the ring? The external opening, that is the spiracle, leads into a cavity, the atrium, which contains a closing apparatus. So the spiracle, so for example, this is the opening. So this opening is the spiracle opening, which opens into an atrium, is what he's talking about over there. So they open into atrium, which contains a closing apparatus. That is, they have some valve-like structures which does not allow the escape of the air to back outside. So once inside, then the atrium is closing over there. So there are these are 2 closing membranes, and their movement can be observed in the sieve plate when it can taken out. When the sieve plate can be taken out slowly, you can see those atrium which opens inside, and the sieve plate prevents dust and dust to enter inside them. The number of tracheal branches start radially from the spiracular cavity. Tracheal branches, we are talking about the tracheal branches over there. So these are the branches that we are talking about. This is the main tracheal trunk, which branches into tracheal bush or the tracheal bushes. The ventral transverse trachea connects the 2 tracheal bushes in each segment, and the trachea ramifies to form tracheoles. So what is this tracheoles over there? So here, if you can see in this picture, so this is the opening of the spiracle. And once the spiracle is opened inside, you can see the main tracheal branch over there. So there are this main tracheal branch, and this tracheal branch forms tracheal bushes. So these are the tracheal bushes that is formed, and this bushes is further divided into tracheoles. So these are the tracheoles over there, something like what we have the bronchioles in our lungs. And the lining of this main trachea has ctenidia over there. These are the segment-like structures. That is, there are some small rod-like structures that you find in this trachea. This helps them to not collapse. So that is when these are all moist, thin layer ones. What happens is when the air is not present, there are a tendency for them to collapse. So the air doesn't pass through. So to avoid that, they have those ctenidium, which helps the trachea to stay solid, doesn't collapse. So that is the importance of ctenidium over there. And When the diameter of the trachea is reduced to 1 micron, these finer tubules are called tracheoles. So these are the tracheoles that we are talking about. And they carry oxygen directly to the cells and the tissues. And the network of trachea and tracheoles, oxygen is carried to various parts of the body, that is the tissue and the cells, and carbon dioxide is removed through the same channel. And as I said, they have the thick ring-like structure called ctenidium, which prevents them from collapsing. Prevents them from what? The trachea from collapsing. Okay. Now coming to circulatory system. So circulatory system over here, we will be talking about how the blood is moving from one place to another over there. So for example, if you see over here, the dorsal vessel, the heart of the insect over here, it has a long tube-like structure called the aorta, and the aorta is opened over there anteriorly, and you have on the heart that is present over here. So this is the long heart that is present over here, and this long heart, you can see there's an opening in each segment. In each segment of the heart, you have an opening, and this opening is called the ostia. So if you remember ostium, we were also talking about this ostium in the porifera, which is the opening for the cavity. A cavity opening is the ostium. So you can also take it in that way. The ostium, that is the opening over there for the blood to flow back into the heart. And the heart is extended posteriorly over here, and the posterior part of the heart is closed, whereas the anterior part, that is the aorta, is open, and the posterior part is closed over here. And the heart region over there, why I kept this picture is these are the aliary muscles that are present beside, that is on the either side of the heart, and these aliary muscles are the one that is going to help in contraction and relaxation of the heart, so that you have the systolic and diastolic pumps that is present over there, the aliary muscles that is present on the abdominal region over there. Okay, so we'll go a little bit deeper into the circulatory system. The circulatory system, as we know, in arthropods, the circulatory system is always an open system of circulation. So for example, what do you mean by an open system? So here you have a heart, and the heart runs into the blood vessels. So they open into the blood vessels. The blood vessels have to open into the capillaries. But what happens over here is the blood vessel doesn't open into the capillary, rather they open into the lacuna or the hemocoel, the cavity, the body cavity of the insects over here. So they just open, they're just released into the body cavity over there, and from the body cavity they enter into the lacuna, and from the lacuna the blood again enters into the heart, so through the ostia that we are talking about. So they have those ostia over there, and through that ostia the blood moves inside, and then again it is oxygenated, and the oxygenated blood is again given back to the surrounding body muscles over there. So dorsal vessel is associated with circulation of the blood and is closely, and this closed posterior is what I was talking about. The anterior portion of the dorsal vessel is finely drawn into a tube called the aorta, and the posterior has the heart over there. Each segment from the second thoracic to the ninth thoracic contain ostium. The ostium is the one where the blood enters back inside. And there are 8 pairs of aliary muscles that is attached. I showed you earlier also. So these are the aliary muscles that are attached over there on the lateral parts of the dorsum. Okay. So this is the picture that you can understand very clearly over here. So here you have the aorta over there. So the pipe in the center is the aorta. And they have these small ones are the heart over there. The heart opens to the ostia, so you can see the openings in between ostia, and they have those aliary 8 pairs of aliary muscles, which helps in the heart to contract and expand, so that the blood is taken in and given out to different parts of the body. Okay, so what is happening in the systolic and diastolic part of the heart? So systolic, as we know, is a contraction of the heart, and diastole is an expansion of the heart. So heart keeps pumping, systolic and diastolic. So what happens over there? When these are affected by, as I said, by 8 pairs of aliary muscles, the contraction and the expansion, the same thing like what we have in the heart muscles, which helps in contraction and expansion. And these aliary muscles function more or less like elastic bands and cause diastole and systole. And as a heart is filled with the blood, a steady wave of contraction progresses in the posterior to anterior region, from posterior region to anterior region. As the posterior region pushes, contracts, the blood that is collected in the heart is moving forward to the anterior region, and from the anterior region, the blood is passed to different parts of the body. So that is what he's saying over there. And hence the blood is discharged to the head and to other different parts of the body. So here you can see very clearly. So you have those the aorta over there on the anterior region, the aorta over there, the head region, and then you have those heart. Can you see the small bulbous heart over there? So these are the heart on each segment, those bulbous, and they have those ostia from where, and also you can see over here the posterior part is completely closed, the anterior part is opened over there. And in this picture, you can see the course of blood. So what is happening is the blood comes out through the ostia and also through the aorta over there into the body segments, and from the body segments the blood is moving towards the posterior side, and then the blood is taken over in the abdominal segments on that. So here the blood leaves through the dorsal vessel, not only by the anterior end but also by the ostia. So here you can see at the thoracic region, that is the mesothoracic and the metathoracic, and sometimes in the first and the second abdominal region also you can find the blood coming out through the ostia, and the rest of it doesn't come out. It is only the uptake of the blood from the different abdominal region over there. But uptake and outgoing is done in the mesometathoracic region of the thoracic region and the first and the second abdominal segment. So this is the course of blood circulation that you can find in the silkworm. Okay, so backward flow of blood is avoided. The backward flow through the body cavity is taken into the heart over there. The blood circulates through the body cavities and appendages, transporting nutrients and removing waste products of metabolism and filling most of the space not occupied by internal organs. So the hemolymph is the one that is going to fill up the place where there are no body organs present. So in the silkworm, the blood cells or hemocytes, that's the cells, do not enter the dorsal vessel through ostia. So they don't enter the dorsal vessel through ostia, but only the blood enters only from the abdominal segment, that is the posterior part of it. Only cell-free plasma is circulated, and the heart pulsates rhythmically, and the frequency of the pulsation varies with the age, temperature, movement, and the race of the larva. So this movement also is the pulsation is also depending on the race of the larva, the species, that is the temperature, and of course the age and the movement. If the movement of the insect is very fast, then the heart rate is also very fast. If the movement is very slow, then the heart rate also is slow in that. So that was about the circulatory system of the larva. Now coming to the nervous system. Nervous system over here, you can see they are divided into central nervous system, visceral nervous system, and the peripheral nervous system. So you have the central nervous system on the thoracic region, that is the head cephalic region, the head region over there. And then you have the thoracic region, the three segments over here, and the abdominal region. So all these thoracic and the abdominal region forms a ventral nerve cord over there. And you have the central nervous system, that is the supraesophageal ganglion that is present. So what is a ganglion over here? Why you call them as a ganglion is in the higher vertebrates, you call the same thing as a brain, whereas in invertebrate, you're going to call them as ganglion because the ganglion is a mass of nerve cell bodies. Say the consists of a mass of nerve cell bodies, that's the neurons that is present over there. And the ganglion has to be placed at different parts so that they are connected. Each and every cell is connected to the brain cell so that the nerve impulse is carried equally to different parts of the body. So here we are going to talk about three: central, visceral, and peripheral nervous system. The central nervous system consists of the brain, which is also called as supraesophageal ganglion. Why supraesophageal? Because it is above the esophagus, so you're calling them as supraesophageal ganglion, which is also called as a cerebrum. The subesophageal ganglion and the ventral nerve cord. So here, this is the supraesophageal ganglion, this is the subesophageal connective, and then this is the ventral nerve cord. Each the nerve cord is again connected to a ganglion, a mass of ganglion, which gives out 2 or 3 pairs of nerve, so that each muscle is innervated, each appendage is innervated by the nerves over there. The brain lies on the head or the dorsal to the esophagus, which is consists of 2 pear-shaped lobules. So here you can see that 2 pear-shaped lobule is what you're going to see over there. And it is formed by protocerebrum, deuterocerebrum, or tritocerebrum. So these are the divisions that you find, the 3 divisions on the dorsal ganglion. And the silkworm larva, the ventral nerve cord, is formed by a series of ganglion on the 4 floors of the thorax and the abdomen below the alimentary canal. So here you can see very clearly how they are differentiated. The larval central nervous system having 13 pairs of ganglia. If you count, you will have 13 pairs of ganglia over there, including 2 pairs on the head region. So you have a 2 pair on the head region over there, the ganglia, and 3 pairs on the thoracic region. So 3 pairs here is the thoracic region, and the rest is the abdominal region. That is the 8 pairs on the abdominal region. And you can see each ganglia giving out a pair, 2 pairs of nerves to muscles and the different segments of the body. So this is the supraesophageal ganglion. And the supraesophageal ganglion is connected by a circumconnective tissue over there, the circumconnective nerves. And this connection is connected to the thoracic ganglion, each one, and then they extend to as a ventral nerve cord, and each ventral nerve cord also has those ganglion on the abdominal region. So they are connected, as I said, by the connectives arising from the posterior and the subesophageal ganglion over there. These are the thoracic ganglion over there, have 1 each, and then they have 1 each on the abdominal segment. Each thoracic ganglion gives out 2 pairs of nerves. So this is one pair of nerve on either side, and this is a second pair of nerve on either side, which innervates the general muscles and also innervate the leg muscles. Why they have to innervate the muscles and the leg muscle is because they have to have that impulse being sent from the brain to the body segments, and from the body segments to the brain. Nerve impulse. Why nerve impulse? Because they have to have that impulses for the contraction of the muscles, and also to tell whether they are on the tactile ones, to touch, the sensory to touch. Okay. And each abdominal ganglion has a pair of principal nerves that innervate the muscles and the corresponding segments. So that was about the central nervous system. Coming to visceral or sympathetic nervous system. Visceral means the nerves going to different parts of the visceral organs is the visceral or sympathetic nervous system. And this is formed by a esophageal sympathetic and ventral sympathetic system. And the esophageal sympathetic system is connected with the brain, innervating the foregut, the midgut, the dorsal vessel, and other parts. So they are going directly to the digestive system over there, the foregut and the midgut. And it consists of a median triangular frontal ganglion above the esophagus. The ventral sympathetic nervous system is formed by a pair of transverse nerves linked with the ventral nerve cord ganglia. A median longitudinal nerve connects each pair of the transverse nerves with the preceding ganglion. So here, this is the ganglion over there. Then they are connected with the preceding ganglion over there. And the transverse nerve innervate the spiracles of the corresponding segment. So the spiracles also have to be innervated by the nerve for the impulse to be sent from to and fro, from the brain to the body part, and from body part to the brain. The caudal sympathetic, caudal means the end region, innervates the reproductive system and the posterior part of the gut, and consists of nerves emerging from the composite ganglion of the abdomen. So that is the visceral ganglion over there. Coming to the last part, that is the peripheral sensory nervous system. It is a fine network of sensory bipolar neurons which have connections with the sensory hair and the central nervous system. And they are multipolar neurons which make an elaborate plexus beneath the integument. That is, what is the function of a peripheral nervous system? They are giving that involuntary action, involuntary contraction and expansion. The contraction of the muscles is all done by this peripheral nervous system with the help of this multipolar neurons. So they give that involuntary contraction and relaxation. So what exactly is involuntary contraction and relaxation is when we are breathing, the lungs involuntary contracts and relaxes. The heart involuntary contracts and relaxes. You don't have to command them, or you don't have to ask them to contract and relax. They do their work. So this is the peripheral sensory nervous system that is working towards the involuntary action of the organism. So coming to the very important part of our topic over here, though the time is up, I'll try to wind it up another 10 minutes or so. So we'll talk about a little bit about this silk glands. What is a silk gland? As we talked about it in the first class, the silk glands are the one that is going to secrete silk, which is going to give us that pure silk, which is of very high value, which is woven into fabrics liked by everyone over here. So here, what are the different parts of that silk gland over here? So this is a spinet that we talked about in the mouth region. There, at the labial region, you have a spinet from where the silk gland is produced. The spinet is connected by a Phillippus gland on either side. The Phillippus gland secretes some kind of fluid substance, though the function of it is not yet known, but definitely they secrete some fluid secretion during the silk gland, the silk production over there. This is the spinet is connected by a pair of anterior silk glands. So they divide over here, and they divide on either side of the gut. So the central part is a gut. On either side of the gut, you have the silk gland present. It is made up of 3 parts over there. What are they? It has an anterior silk gland. Then this is the middle silk gland that is present over here. So the yellow color, the orange yellow color is the middle silk gland that is present over here. And the red part is the posterior silk gland. So this is the one that is made up of over here. The posterior silk gland is most important because this is the one that is going to secrete, produce a protein called fibroin. So this is a fibroin, and you can see the structure over here that is present inside this posterior silk gland. The posterior silk gland is most important because this is the one that is going to produce that fibroin, and the fibroin is the one that is most important in the production of the silk. So that is a main silk that we are seeing, and it is very strong and rigid. And around this fibroin is sericin that is produced, and the sericin is produced by the middle silk gland. The sericin is a covering for the fibroin, but the important part of the silk that we are utilizing is nothing but the fibroin that we are forming them into fabric. Sericin is only a protective layer around the fibroin. Okay. So silk glands transformed labial glands. If you remember the labial gland that we talked about in the first slide, where in the head region over there. So here you can see, so this is the mouth part of here, and the labial glands are transformed into an ectodermal origin, which is cylindrical and tubular with a characteristically branched nuclei over there. And it is the second largest part of the body, and each gland is several times the length of the larva, occupying the 2/5 body part. So for complete 2/5 part of the body is covered by the silk gland, situated on the 4th to the 8th abdominal segments, and they are present at the ventrolateral sides of the body. And anteriorly, the paired ducts unite to form a spinet. So this is a spinet, which is secreted, which helps in secretion over here. Okay, so you can see they are divided into 3 parts: the anterior, the middle, and the posterior part. The anterior part opens into a tube-like structures in the foregut. The middle part is the largest one, which is having again divided into 3 regions, and that is the anterior region, the middle region, and the posterior region. And the beginning of the middle region is narrow. The posterior region is crooked and curved between the dermovisceral muscles. A pair of glands known as Phillippus, as I told you, they secrete a viscous fluid secreted by these glands, and the wall of the silk gland is composed of 3 layers: the tunica properia, the gland cells, and the tunica intima. And this is how the spinet looks like towards the end. The sericin, that is the middle region of here. This is the anterior middle region. This is the middle region over there, and this is the posterior middle region over there. And the middle region one is one that is going to secrete the sericin, and the posterior region of the silk gland is the one that is going to produce the fibroin, as I already told you. So these are what I have already told. So you can just go through these slides over there. And as I said, the silk fiber is made up of inner fibroin and outer sericin. Okay, so this is what I was talking about, fibroin and the sericin over there. So coming to the reproductive system of the larva. So here we are going to divide them into larval reproductive stage and the moth reproductive stage. So here what you can see that in the segment, that is when you talk about the larval forms over here, in the 8th abdominal segment, if you can see over here, there is one foregland called Ishvita's gland that is present on the 8th abdominal segment. And on the 9th abdominal segment, you have a hind gland, Ishvita's hind gland. This is the name of that gland that is present over there. And in the male region, the male part on the 8th and the 9th segment, between the junction of the 8th and the 9th segment, you have an Herold's gland, a protrusion like white color structure that is found. So this is how they are differentiated between male and a female larval stages over there. And when you're talking about the female moth, that is the adult reproductive system is what we are talking about. In the adult reproductive system, if you can see in this picture, they have this bursa copulatrix. So this is a small bursa copulatrix, and they open into the ovipositor over there. So this is the ovipositor. The ovipositor is where which helps in laying of the eggs, positioning of the eggs, because as I told you, the eggs are positioned clearly as a round circular motion. They move in a round circular motion, and the eggs laid very cutely in a round motion. So that this is helped by this ovipositors over there, and they have the ovarians. So this chain-like structures is what is the ovarioles. That is, the ovary is segmented into ovarioles, each giving rise to single eggs over there, and they are alternated by a cell which is going to take care of each eggs over there. So they're going to nourish the egg and a nourishing cell, egg and a nourishing cell. So they have an alternate egg and a nourishing cell. And each ovarioles is made up of 4 branches over there. Can you see 1, 2, 3, 4? So each side they have those 4 branches, and each one is going to position their eggs. So you can see the eggs coming out over there in this picture very slowly, lightly over there, and through the ovarioles, the oviduct and the ovipositor, you can see they are being dropped down. So this is the female reproductive system over there. And at the time of mating, the oedicus is received by the ostium bursae. So the bursa copulatrix opens into a small ostium bursae over there, a very small ostium bursae, where they take up the oedicus. That is, the oedicus is the male part of the reproductive system, and the oedicus secretes the sperm, which is lodged inside the ostium bursae. So if you can see over there, the ostium bursae, and they have this spermatica over there where the sperms are being stored. I'll show you a better picture over there. So this is the better picture of the female reproductive system over here, if you can see. So these are the ovarioles. You can see the eggs that is formed inside these ovarioles. That's the ovary. And then they have the spermatica. So this is a spermatica where the sperms are lodged. That is, once the sperms are lodged inside the female reproductive organs, the sperms are lodged over here in the spermatica. And from there, they are released into the egg for fertilization. And this is bursa copulatrix, and ostium bursa is present over here where the oedicus lodges itself inside this female reproductive organ, and where the sperms are lodged, which is taken by the spermatica and taken care of them. And these are the accessory glands to take care of the eggs development over there. And they have this common oviduct and the ovipositor where the eggs are being laid. And these are the sensory hair of the female reproductive organ. So coming to male reproductive organ over here. So if you are going to check out the male reproductive organs, so these are the testes on either side, the kidney-shaped structure, something like that. But these are the testes over here. And the testis opens into vas efferens, and the vas efferens opens into vas deferens, and the vas deferens opens into seminal vesicle, and the seminal vesicle opens into an ejaculatory duct over there, and from the ejaculatory duct they open into edicus, and this is the edicus is the one that is lodged into the female. So this here you can see very clearly. So these are the accessory glands. They have those testes over there, the flower-like structures, the testes. Testes opens into vas efferens, and the vas efferens opening into deferens, and the deferens opening into seminal vesicle over here. The seminal vesicle where the semen is lodged over there, that is the sperms and the semen liquid. This opens into an ejaculatory duct, which opens into edicus, and this is from where the sperms are released into the female. So this is the reproductive organ of the moth. Coming to embryogenesis over there, embryogenesis is divided into different parts of here. That is after fertilization, that is after hatching, the fertilized eggs undergoes cleavage, and after cleavage they undergo blastodome formation. Then you have a germ band formation. Then after germ band, organogenesis occurs. Organogenesis where the organs are developing inside them. And as the organogenesis is occurring, blastokinesis occurs. Blastokinesis is reorientation of the larva inside. And you also have the eye spot or a blue egg stage formation. And once the egg comes to an blue egg stage formation is where hatching occurs. So this is what we are going to talk about very quickly over here. So here you can see the cleavage. After the cleavage, you have a blastodome formation and the germ band formation. That is, the cleavage proceeds through a succession of mitotic division of the zygote, which undergoes blastodome formation. So you can see the blastodome occurring on the ventral surface over here. As the blastodome is formed, they move on to the next stage called the germ band. The germ band is nothing but you can see a band-like structure on the egg cell over there. So this band-like structure is called as a germ band. So this germ band undergoes different formation in this 24 hours. That is on the ventral side of the egg, they divide into lamelliform structure called the ventral plate. The ventral plate further thickens to form a germ band called the daruma stage. And the membranous layer forms two folds around the germ called serosa and amnion. So this is the germ band stage over there. And the germ band undergoes the growth of the embryo. So here you have, as the germ band grows, the cell division occurs. That is, they form three layers. That is the ectoderm, mesoderm, and the endoderm, because they are triploblastic. So they have the three germ layers, the ecto, meso, and endoderm over here. So here you can see the seventh stage. They have undergone gastrulation. The germ band undergoes gastrulation. And the gastrulation moves on to formation of stomodium and the proctodium over there. Which undergoes the segments over there, the stomodium and the proctodium, they form into head structure. So you have the head region and the thoracic region over there. So this is the head region and the thoracic region formation. And they undergo blastokinesis. What is a blastokinesis? There is a reorientation of the larval form that is from the ventral side. They come onto the dorsal side. Is called as the blastokinesis stage. And as a blastokinesis stage proceeds, you can see that the larval formation occurs over there. So you can see a clear picture of the larval formation occurring. And as the larva is completely formed, the larva undergoes hatching is when those small ant-like structures comes out of the egg. So here, the blastokinesis, what is the important point you have to remember? What is blastokinesis? The active movement of the embryo from the ventral to the dorsal side of the egg. And at the same time, they undergo a 180 degrees axis movement is what is blastokinesis. Then we talked about the eye spot stage. So what is an eye spot stage? Bristles grow around the body surface. As I said, bristles, they help in the growth of the larva over there. The dark pigmentation around the mouth and the head can be seen through the egg. So you can see over here very clearly. So this is the blue egg stage and the eye spot stage. Can you see the dark dots? So this is an eye spot stage. where it is ready. So once the egg reaches that eye spot stage is where you can say that the eggs are ready to hatch. Now you can compare, the other eggs don't have those, so they have not undergone any development over there. So these are the ones. So those that haven't undergone the development, you can also call them as atreus cells, where some of the cells, they don't develop into yolk at all. So that is one type of eggs, which are you can also call them as non-polar bodies sometimes, like, yeah. So blue egg stage, the whole body of the embryo turns black due to the development of the pigments. So here you can see over there. So this is the black pigment that you have seen over here, appears bluish black. Egg should be cold stored. So if you want to take care of them, then you have to cold store them. And hatches within 24 hours after the blue egg stage. Embryogenesis ends when the yolk contents have been consumed. That is, the larval form is consuming the yolk content that is present in the egg. And the immature insect is fully formed and ready to hatch from the egg. So here you can see they have been hatched from the egg. So yeah, so that was about the silkworm and its different stages of its life cycle. And here you can see in this picture, you can see the different colors of the larval forms and different colors of the pupa and the moth stages of here. So this was all about the silkworm biology. And I hope you have understood it. And if you have any doubts, you can get back to me during the class hours. So, okay, students, that's it for today's class. Thank you.